Well, I'm going to start indicating that this presentation is in some way follow-up in relation with our project, our intra-CIIIEN project. So what we think is that we have some duty with all members of CIIIEN, showing our results in the context of intra-CIIIEN project. So this is not a finished project. And what I'm going to do is to show you what we have done with our money in some way. So some of this work has been published, other work is in preparation, and other work is going on. Well, the topic is the involvement of neurons and astrocytes and oligodendrocytes in seeding and spreading in neurodegenerative diseases. And you see that many, the main aspects of neurodegenerative diseases are indicated by the presence of some abnormal deposition of proteins in nervous cells, glial cells, and other structures in the brain, and are characterized by the certain regional and cellular vulnerability, and also by the progression of the disease, as it here exemplified by the different stages of tau in Alzheimer disease. Well, our idea is that the progression of this disease is mainly related with the capacity of seeding and spreading of the abnormal proteins from one cell to another cell, as it has been perfectly demonstrated and identified in prion diseases. So our model is that of prion diseases, in which we can inject some inocula in the brain of animals, normal animals. The inocula is composed of, or is formed by abnormal PRP, and then we can see in these animals the spreading of this abnormal protein within the context of the new animal, which was devoid of this abnormal protein, but which has the normal protein to be transformed. Well, in other diseases, the term seeding and spreading has been developed in the last years. So seeding in other neurodegenerative diseases has been reported for beta amyloid, for hyperphosphorylated tau, in alpha-synuclein in Lewy body diseases, and in multiple system atrophy, TDP-43, SOD1 in ALS, and Huntington disease. The majority of these experiments have been carried out in mouse models, in which which are expressing either normal human protein or mutant protein. For example, in cases with, or in the study of tau, the majority of models are animals in which human tau or human mutant tau is expressed instead of murine tau. The conditions and the characteristics of the properties of the seeding and spreading are exemplified by different models, showing that some of them, this kind of transmissions can be carried out by synaptic transmission, other by nanotubules, other by cytosis, and several different mechanisms. What is important is that it seems to be necessary a direct cell-to-cell transmission through, or perhaps the extracellular space, and that there is an exponential progression of the biological processes. Regarding tauopathies, in which our study has been centered, we know that there are many different tauopathies. The best known is Alzheimer disease. This is a 4R, 3R tauopathy, but there are many other tauopathies in which there are deposits in neurons and glial cells, and the main abnormalities based on the deposition of 4R or 3R tau. Among them, we know Alzheimer disease. We also are going to study which is part of Alzheimer disease. That means patients or cases in which there are neurofilament tangles without beta amyloid, aging-related tau astrogliopathy, which is a pure astroglial 4R tauopathy, The other one is 3R, 4R. Next one is globular glial tauopathy, in which is a very characteristic tauopathy with involvement of neurons, astrocytes, and oligodendrocytes with very typical and specific inclusions. And then other tauopathies as PSP, progressive supranuclear palsy, peak disease, the later characterized by the position of 3R tau. As I have said, there are many different deposits in these cells. So in neurons, we can consider, for example, pretangles, tangles, and in particular diseases, peak disease, the characteristic peak bodies. In astrocytes, regarding astrocytes, we have plaques, astrocytic plaques in corticobasal degeneration, thorn-shaped astrocytes in ARTAC, and in cases of aging. And, for example, toothed astrocytes in PSP, and globular astrocytes and oligodendroglial cells, GGA or GOA inclusions. And regarding oligodendrocytes, we can see coiled bodies in the majority of cases, and particular oligodendroglial inclusions in globular gliopathies. Well, the objective is to study what happens after following the injection of the inoculation of soluble and insoluble fractions, sarcosyl insoluble fractions, into the brain of mice with expressing murine tau. Together with the analysis of the different tauopathies, the different conditions on the different spread of the deposition after the inoculation, the cell types which are involved, and the problem mechanisms which are involved in this process of spreading. We have used sarcosyl insoluble fractions together with sarcosyl soluble fractions of a large number of different tauopathies, 4R, 3R, primarily neuronal, primarily astrocytic, combined, and 3R, 4R, to know what happened with different particular strains, tau strains in the different models on the different diseases. And to use this kind of murine tau model, expressing murine tau, to know what is the part of the response, or the responsibility of the response regarding tau spreading and seeding in relation with the characteristics of tau in the host. Well, you can see here what is the characteristic of the main compound of the different inocula that we have. I'm sorry. Well, I'll put it here. The different sarcosyl insoluble fractions. In this case, the difference between part, ARTAC, GGT, PSP, PAD, and one frontotemporal degeneration linked to this kind of mutation. As you can see here, the pattern of phosphorylated tau is different in all these diseases, in the different diseases. And some of them are characterized by 3R, 4R, or 3R, or 4R tau. In addition, by using electron microscopy, we can see the difference between or among the fibrils and the different conditions. And here is a theoflavine test to show the amyloid or the trapping of staining of theoflavine by the different inocula. And you can see here, for example, AD, GGT, and ARTAC have this typical curve, this one, whereas the soluble fractions have flat patterns, indicating lack of amyloid characteristics. Well, what happened following unilateral inoculation in the hippocampus sarcosyl insoluble fractions in these different conditions? So regarding Alzheimer disease, we can see following a survival time of 6 months in the hippocampus, we can see here the presence of staining key neurons in the fimbria, and also in the ipsilateral, middle, and contralateral part of the corpus callosum. Here is another picture observing the alterations following part in which the pattern is more or less the same neurons in the hippocampus, in the fimbria, glial cells, and here glial cells in the different parts of the hippocampus. And here another important point is that these cells are 3R, 4R as expected, and they contain also abnormal tau, tau abnormal conformed, but not truncated tau as is observed, for example, in Alzheimer disease or in part. Regarding our TAC at 3 months, we can see in the hippocampus here and in the fimbria, whereas following a survival of 6 months, the position is also found in the other part of the hemisphere, as well as certain group of neurons in the septal nuclei and in the periventricular region. And again, this is a very important point, is that in addition to 4R tau, as we expected for our TAC, which is a 4R tauopathy, also these cells contain 3R and tau in the CIIIEN region and in the dentated gyrus. Well, the next one is an indication of what kind of structures are we labeling. And some of them are in part are neurons, not only neurons, but cell processes also, distant processes, and also cell processes in the different regions, in distal regions. So indicating differences between what we observe following 3 months and what we observe following 6 months. And using double labeling immunohistochemistry, we show that there are neurons and mainly oligodendroglial cells. And this is a very important point because, as referred before, astrocytes and oligodendrocytes are in some way forgotten in the context of neurodegenerative diseases. Well, and what happens with our TAC? With our TAC is almost the same. There are neurons, there are astrocytes, as expected, and oligodendrocytes here by using double labeling of different markers, cellular markers. However, microglia is never colocalized with abnormal deposits. And what is curious here is, although our TAC is a pure tau astrocytopathy, here there are neurons which are also labeled. And another important point, and we will go later on, is that these cells that contain 88 tau, phosphorylated tau, also contain active kinases, as for example this one, P38. Well, this is the tau spreading the seeding can spreading following inoculation in the part hippocampus following 7 months. We can see the spreading through to the other part of the brain and in different distal regions. Here is our TAC at short intervals, 3 months after survival. Just in the ipsilateral hemisphere, but following inoculation and survival of 7 months, we can see the spreading to the contralateral hemisphere and exactly the same regions as observed in 4 part. Here is GGT, the other one, and is exactly the same. So we inject in the hippocampus, and then we observe neurons positive neurons in the hippocampus, in the fimbria, in the ipsilateral middle region and contralateral region of the hippocampus. And again, we see 4R and 3R. So this could be expected just to be a 4R. And when we are analyzing by by double labeling immunofluorescence and confocal microscopy, we see that neurons and many oligodendroglial cells are the main targets of this inocula. And what is important in this case is that in the same way we cannot see reproduce thorn-shaped astrocytes in our TAC, which are characteristic of our tag. Here we cannot reproduce the global glial inclusions, astrocytic or oligodendrocytic, in following the injection of GGT. What happens when we inject in the corpus callosum? This is a very important point because the majority of tauopathies have involvement of the substantia of the white matter, and then we need to know what happened in the white matter, whether it is a primary alteration of the white matter or not. And then the inoculation in the corpus callosum is very useful to demonstrate that following inoculation, the tau deposits go to the are in the ipsilateral, the middle region, the contralateral, either following inoculation with PART, with RTAC, with GGT. Also, there are subtle differences between or among the different tauopathies, and the same happens with GGT. I'm sorry, that's that. PART, RTAC, GGT. Well, GGT almost is the same, and here, following PSP, following peak disease, and following frontotemporal lobar degeneration due to mutations of this kind of mutation, P301L. And following GGT inoculation, what we observe is exactly the same pattern. So what is important in the present case is that, or in the present context, is that the oligodendroglia, in addition to threads in the wet matter, are the main targets of tau deposits in these models. And here is the spreading, the seeding and spreading in relation with the timing. So we can see the spreading to the contralateral side. And here is the observation that there is associated with the activation of certain tau kinases, so indicating that this is an active process. And although this is not accompanied by cell loss and apoptosis, there is an alteration of the myelin in these animals. So the involvement of the oligodendrocytes has a negative effect on the production of myelin, as revealed by PLP1 immunohistochemistry. Here is the alterations in the corpus callosum, and here the differences between human samples and differences between and the animals. And what we can see here is that there is not an exact reproduction of what happened in the human brain when the model is the mouse. In summary, what we have seen is that these are just the things that I mentioned before. So what is interesting is that there is not an exact reproduction of what happened in human diseases. 4R3R tauopathies show the same pattern. And what is interesting in this way is that probably the connections or synaptic connections are important in tau seeding and spreading in neurons, between neurons, but probably this is not the mechanism regarding oligodendrocytes. On the other hand, oligodendrocytes are main targets of the main targets of these tau deposits. And we see here that in addition to different species of tau or strains of tau from donors, this is also very important what happens with the host, the tau in the host. So it's in some way what has been reported previously regarding the prion diseases. So there is some kind of brain barrier. The last one, when we use newborn animals, then we cannot reproduce this kind of alteration. So at the end of the 6 months, there is not deposits. When we inject it in other regions, what happens is different vulnerability. So, for example, the striatum and the thalamus have a lower capacity of spreading of tau, but there is a different, again, with age. So when animals are injected at the older ages, they are more vulnerable to tauopathies, as can be expected from animals. And in the end, this is just a summary of these results. These are, in the end, preliminary results. What we have seen also is that when we inoculate or re-inoculate animals which were inoculated at the age of newborn, these are not protected at all. And we think that in addition of these mechanistic aspects of seeding and spreading, this is also an important point, which the present models are probably very useful in the study of therapeutic strategies in the future. And this is the second part of our project. And this is our acknowledgement in relation with our group, also members of our group, together with José Antonio Río, Jesús Ávila, which are members also of this intra-cybernet project, and also this work was funded by different institutions and conditions. And thank you very much for your attention. Thank you.
Thank you, Isidro. We have time for one short question.
Great talk. I was wondering whether you have tried to see whether this pattern of spreading would be the same in mice that have humanized tau, that they have removed the endogenous tau and they produce the whole genomic. I know there are some, but I don't know if they are good enough for the experiment. I think it's important because you mentioned at the beginning the importance of the ratio of 4R versus 3R tau, and mice express much more 4 tau than 3R tau, unlike humans that they are in normal conditions similar amount. So maybe it would be nice to see whether the patterns of spreading that you see in a wild-type mouse would have changed if they have the humanized version of the gene tau.
Yes, we agree with you, and in fact the project also covers the study of humanized tau. So this is the other part, but I think that here was important to show that wild-type animals have been reported that they are not vulnerable to this kind of tau spreading and seeding, or that these animals show the same pattern as the others showing humanized tau, and this is not true. So there is an important proof of concept in this kind of things. So what we are trying to demonstrate is that...
Well, just to make sure that the pattern of spreading would be the same in
yes,
both
it's
conditions.
not exactly the same when using humanized tau, and authors have said that we can reproduce toothed astrocytes and etc. We are not sure about the results because the pictures are not very clear. But in any way, what we are intending to understand is why in some tauopathies there is an involvement of certain cells, certain neurons, or certain glial cells, etc. And this is probably not only due to the fact that it's the host, the tau, the strain of tau which is abnormal, but also the tau in the target cells, which is also different. So it depends on both things.
Okay, thank you. Thank you, very nice talk.